Welcome back. In this video, we're going to look at our second line of defense. So this only comes into play after failure of our first line of defense where pathogens have breached those external barriers. And there are several different components to this. And the first part I want to review is the role of specific blood cells in the tissue, what they can do as defense. So starting off first, our neutrophils, our first responders, when they wander in connective tissue, they are going to do phagocytosis and they are going to digest single cells. And that's fine if they only find here a bacteria, there a bacteria. But if there is a lot and they recognize there is a lot of foreign material that they need to get rid of, they have a more robust method of attack. And that is by releasing their granules and creating a chemical killing zone all the way around them. And it kills all these cells, both good and bad nearby, and it also kills the neutrophil itself. But this is much more effective than individually going and killing one pathogen at a time. In contrast to that, eosinophils are not going to be found in our skin, but they go to our mucous membranes inside your nose, inside your mouth, inside reproductive systems, anywhere where you have epithelial lining on the inside. And so they're really prominent at sites of where allergens have crossed the barrier, or you have parasites, or you have an inflammatory reaction that has been around for a little bit. They will also release their granules and cause a chemical killing zone. And their chemical killing zone also does a couple of other things. It helps break down antigens, antibody reactions. So if an antibody has recognized something that's foreign, it will destroy that. And one of the very interesting things that eosinophils can do, and if you remember, they are generally anti-inflammatory, they actually promote basophil action, and basophils are pro-inflammatory. So it seems kind of counterintuitive, but they limit the amount of basophil action that can happen, whereas basophils not under the influence of eosinophils have a much more robust response. So speaking of basophils, I just want to remind you that when they move into tissue, we change their names and we call them mast cells. And mast cells are important because they secrete these cytokines known as leukotrienes. These are chemicals related to prostaglandins that activate and will attract the neutrophils and the eosinophils that we've just talked about, thereby increasing the inflammatory reaction. And they release their chemicals inside the cell. So remember, one of the two big things that they had was histamine. So the histamine is pro-inflammatory. It can be limited by eosinophils, however. And the other chemical we talked about that they have was heparin. And heparin is a compound that blocks some of the factors required to form a blood clot, not a platelet plug, but an actual blood clot. And the reason is we don't want huge blood clots to be forming, because blood clots will block the flow of blood. And if you block the flow of blood, that means any white blood cell here cannot get down to there. So those are the effects of our 3 granulocytes. Looking at our 2 types of agranulocytes, the first are our lymphocyte-related ones, the lymphoid stem cell line, which are our natural killer cells, our B cells, and our T cells. And I'm not going to talk about them right now because we are going to have 2 full Zoom lectures on their effects. So that leaves me with the monocytes. And so far we've learned that monocytes in tissue become macrophages, and so they do phagocytosis. And some of them are stuck in certain locations for their phagocytosis, but others can wander around and seek out pathogens. They don't have to wait for the pathogen to come to them, and that's a little bit more effective. But monocytes can become more than just macrophages. Something else monocytes can become are dendritic cells. And dendritic cells are in multiple different locations in our body. But in our skin, dendritic cells we call Langerhans cells. And remember, the Langerhans cells were like the Roomba vacuum cleaners cruising throughout the various strata of the epidermis. And dendritic cells are cool cells in that they look kind of star-shaped. They have all these long processes. And when they come across an antigen, they will phagocytose the antigen and chop it up. And then they actually present the antigen so that it will activate a T cell, as we will learn about in a subsequent Zoom video. And so this little square in the bottom right-hand corner is just so you can recognize the differences between macrophages and dendritic cells, even though they come from the same monocytes circulating in the blood. So that's it for the cells. I have made a little table for you, including cells that we're going to be talking about in the third line of defenses down there in the bottom. So once you have done everything, come back and look at this and see if that helps clarify and simplify the material for you. So let's get started with our other non-specific types of immunity. And the first thing, remember, we're in the second line of defense, but we're still non-specific for both the first and second lines of defenses. Whatever it is has broken through the barrier. And in inflammation, this is a response that kind of takes you further away from normal, but then it brings you back to normal. And there's a lot of things that are going on in the inflammatory response. So what I want to point out is in a normal person, it will take 7 to 10 days for a normal inflammatory response. It usually peaks at around 48, 72 hours, and then it returns you back toward normal by about 7 to 10 days. So that's an acute inflammatory reaction, and that's what we're going to be talking to. So if you stopped and think, when you twisted your ankle, that was an example of an inflammatory reaction. And so it got red, it was hot, it was swollen, and it hurt, and you limped, okay? Those 5 things are the 5 cardinal signs of inflammation. And in Latin, they rhyme, but I'm just giving them to you in English. So if you think about your twisted ankle, you usually can hit all 5 of these. There is redness, swelling, heat, pain, and loss of function. And so we're going to go through the various steps of inflammation. As we're going along, I want you to realize, oh, that's what causes the warmth. Oh, that's what causes the redness. Oh, that's what causes the swelling. And I'll just tell you right now, the loss of function has to do with the pain. All right. So starting with this, in this example, you have poked yourself with a pin and introduced bacteria into your tissues. Now, in addition, I want you to realize when that pin went into your skin, it did not squeeze through all the cells and not damage any of them. It damaged cells, and those damaged cells are releasing those chemical signals, those cytokines, some of which are going to be chemotactic factors. So we start with tissue injury. One of those factors that's released by damaged tissue is histamine. Yes, the same stuff that it's found in basophils, but you don't have to damage a basophil, because remember, basophils are in the tissues as mast cells, and those mast cells will have histamine. So histamine has been released, and an interesting thing that histamine does that we haven't talked about, it actually causes the local blood vessels right around where the histamine concentration is to vasodilate. So what that means is if we had endothelial cells which are right next to each other, when that blood vessel vasodilates, we get tiny little spaces between those endothelial cells. Okay. And so the dilating of the blood vessel will increase blood flow into the area, making it warmer and changing the color. Now, we also are going to have chemotactic factors being released at the same time, and histamine promotes that chemo attraction to happen. So because the blood vessel has vasodilated, it now has become leaky. It has what we call vascular permeability, meaning fluid inside the blood vessel can leak out. And not only that, our white blood cells can undergo diapedesis, which means now they can move to the site of injury. And we have extra fluid in the site of injury, making it increase in size. In addition, if a blood vessel was injured, the platelets would be activated and they would start that whole process of coagulation. Now that we have white blood cells in the tissue, remember the first responders are going to be neutrophils, and so we're going to have a lot of neutrophilic-based phagocytosis for the first 2 or 3 days. We are going to have more monocytes entering the tissue, becoming tissue macrophages, and that will start significantly going up after 3, 4, 5 days. And it will continue for as long as needed. And so both of these cells are going to do phagocytosis, not only of any bacteria that were introduced, but also of any debris that occurred as a result of the injury, any cellular death that occurred. Now, in addition to this, there's an entire process of tissue repair with collagen deposition and scar formation that occurs, whether it's on a microscopic or a macroscopic level. But we're not going to talk about this. Nursing students, you will learn a lot about this in your first semester of nursing school. So let me just review that white blood cell diapedesis for you again. Remember, it happens at a site of injury where we are going to have damaged cells releasing inflammatory chemicals. Now, causing the vasodilation. So in the blood vessel itself, what happens is the endothelial cells of the blood vessel will release these molecules, which are called cell adhesion molecules, because that's just telling you what it's going to do. What it does is it makes the surface of the endothelial cell sticky, kind of like the back of a Post-it note, sticky. So when white blood cells are slowly tumbling around, they're going to stick to this, and then they kind of like walk along the surface of the endothelium until they can squeeze between gaps in the endothelium and set up a new colony at the site of injury in the tissue. So here's a slide which, instead of having 3 separate pictures, puts it all on one. So if you want to pause the picture and talk yourself through the entire process, that would be a great thing for you to do. All right, fantastic. Now that you know our first non-specific response, which was inflammation, I want to teach you about our second one, which is the complement plasma protein system. So when we talk about activating complement, this is what we are talking about. So what this is a group of proteins that are floating around in your blood all the time, okay? And they don't do anything unless they get activated. And once one gets activated, the fact that it is activated will activate another protein in the system, which then can activate another protein in the system, and so on until we get to the final product being produced. So I want to mention that there's actually 3 ways to activate complement. Excuse me, the classic one is if we have antibodies which are binding to antigen. Just the fact of that will come back and activate this. Another way to activate it is spontaneously, okay? And we're not going to talk about that, and we're not really going to talk about any way that we can activate it. But the third way of activating, there's certain pathogens which have certain sugars on their surface, and just the presence of those activates this. So I want you to realize that this is happening all the time. I remember when my youngest son was a freshman in college, like at 2 o'clock one morning, he goes, "Mom, why didn't you ever tell me about complement?" And I go, "Why would I tell you about complement?" He goes, "I spent an entire year of college learning college level biology and nobody mentioned complement." And I go, "Well, then you had a sucky bio teacher." What can I say? And it's just like, it just freaked him out to realize that his body was doing this 24/7, keeping him healthy, and nobody had ever mentioned this because it's a really cool system. All right, so let's look and see what happens. So it doesn't matter how it's activated. What is going to happen, no matter how it's activated, eventually we produce what's known as C3. So the C stands for complement. So this is like the third molecule, okay? So C3. Now the interesting thing about C3, it's going to split up into what we call C3A and C3B, which have totally different roles from each other. C3A actually goes back and triggers histamine to be released from mast cells and from basophils, thereby turning the inflammatory reaction on. And if there already was an inflammatory reaction, then it turns the burner on higher. In contrast to the histamine effects of C3A, C3B doesn't care about monocytes, but what it cares about are your neutrophils and your macrophages that can do phagocytosis. And so it's going to turn the burner up on your phagocytes so you have more effective phagocytosis. So hopefully you can clear all the bad stuff. So they are doing that. Now, in addition, whether you went down the C3A pathway or the C3B pathway, eventually they come back and activate C5. And C5 is important because C5 is going to activate C6, which will do C7, which will do C8, which will do C9. And this complex is called the membrane attack complex. So just looking at its name, what do you think the membrane attack complex is going to do? Yeah, it's going to attack the plasma membrane of something else. So let's look and see how that does. So remember, we're still a non-specific, okay? So here is the phospholipid bilayer on a pathogen, okay? C5 comes and attaches it to itself, and then C6 comes and C7 comes, and then we have C8 come along, which actually is able to poke in all the way transmural, and then C9 comes along. And so what is happening? C9 pokes a hole, another C9, another C9, another C9. And if you notice, the C9s are going all around and forming a circle. And what do you think happens when you have something poking all the way through the plasma membrane in a circle? What is going to be the net result of that? That's right, you're going to have holes, okay? And so here is a very nice discrete hole that you can see on the side, because you can clearly see the complement proteins up here and the hole in the plasma membrane. But now looking from above, I want you to realize you got lots of holes in this pathogen. And as a result of all these holes in this pathogen, water and sodium are going to rush into that cell, and that cell will get bigger and bigger and bigger, just like your balloon, until it bursts. Okay, so basically we say the membrane attack complex, and that's what they're abbreviating MAC here, is what causes death of that antigen cell. All right, so isn't that kind of a cool thing? All right, so I've talked about pathogens, and let me explain pathogens. There's more than one kind of pathogen. So sometimes we're talking about viruses, for instance, measles or hepatitis or coronavirus, okay? Sometimes we're talking about bacteria, such as with strep throat or with this cute little organism I put up here. This is what E. coli looks like. It's kind of cute, so that's why I like to nickname it E. colita. So if you don't want E. colita, then please do not eat any hamburger that is pink. Your burgers must be well done or you're at high risk for an E. coli infection. In addition to bacteria, we also will have fungi. So down here, this is showing an example of athlete's foot. We can have single cell little organisms known as protozoa. This is what causes malaria. Or we can have our other parasites, such as various worms that you can pick up when you are they're not common in the United States. I'll just leave it at that. So I want to talk about viruses because students tend not to have had much biology. So you know the word virus, but you don't really know what a virus is. Okay. First thing, it's a group of chemicals. It is not alive. It is not a cell. So when you hear them talking on TV about coronavirus and they talk about, well, it's going to kill the coronavirus. Okay, the coronavirus is not alive. It's not a cell. Okay. And so as long as it stays like chemicals in a jar on a shelf of a laboratory, it can't do you any harm. It has to get into your cells. And I'll talk about how that happens. But first, what is a virus? A virus, basically, in its most simplistic form, is a nucleic acid inside a protein coat. That's all that is required. So we have this, what we call a capsid, which is made of proteins, inside of which we will find a nucleic acid. And viruses will either be DNA-based or they will be RNA-based. That's all that's required. Now, many viruses are much more complicated. I know you've seen the little pictures and you're going, I don't understand what all those projections are. It's called coronavirus because the little thing sticking out from the protein capsid look like a crown corona. Not crown beer corona. Okay, so how does it reproduce? For this, kind of think of viruses like the pirates. So let's bring Captain Jack in. So the first thing a virus has to do is it has to get inside a cell. So it commits a crime, as pirates are wont to do. And the crime in this case is breaking and entering. It's got a couple of steps to it, but that's not important for our story today. Once it's inside the cell, it's going to hijack the cell, as in cell, I don't really care what it is you wanted to do, but I'm the boss and you're doing what I want. And what is it I want again? Oh, oh, me! Make a bunch of me. I'm a lot narcissistic, okay? And so it hijacks the cell machinery, AKA the nucleus, and says, make a bunch of me. Put your finger on the Xerox machine, and what I want is a whole bunch of me. And then once the whole bunch of me is made, we all escape, and we go and we look for another cell/ship to break and enter into. All right. So what does that look like if you look at pictures in a science book? It looks more like this, which is not nearly as exciting a story, but I'll tell it to you anyway. Start here in the lower left corner of this, and this is a virus, and this actually happens to be an RNA virus because this is single strand in purple, and it's got protein coat on the outside. So it now in the second picture, it has entered the cell. It's going to be releasing its viral RNA, which then goes into the nucleus, and the viral RNA hijacks the host cell DNA. The host cell DNA makes the usual messenger RNA, does the transcription translation thing. So we have our ribosomes on our transfer RNA, and now we're making long strands of protein, which basically are viral RNA, and it's also making the proteins for the protein coats. And so what happens is the new viral RNA basically buds from the surface, and it has protein coat from the cell, and now it's off to create more havoc elsewhere. So what does that have to do with the second level of defense? Well, interferon. So we already had our cells we talked about, then we talked about the complement plasma protein system, creating membrane attack complexes. Now, our third thing we're going to talk about is interferon. So I just kind of want you to listen to me talk you through these pictures. And we're going to start, this is a cell on the left, and the purple part is its nucleus. So here's a lovely little virus with its nucleic acid. It enters the cell, it drops off its nucleic acid. Yes, it's going to hijack machinery. We're going to make the viral particles. It's going to escape, and it can infect another cell. That's not the part I want to talk about because I've already done that twice. What else happens is when the viral nucleic acid enters the nucleus and becomes part of the host cell's DNA, not only is that host cell reprogrammed to make new viral particles, but it also is programmed to produce a molecule known as interferon, which are these peachy triangles. So when this cell dies, because it dies because the virus basically has killed it, then the interferon is going to be released by the cell that gave up its life and produce a whole bunch of viral particles. So what's the whole point of this interferon? Well, on the outside of cells, there are receptors for interferon. And when the interferon binds to that cell, that stimulates genes in the nucleus of that cell to create antiviral proteins. So if these antiviral proteins are made before the new virus enters that cell, that virus can't replicate. On the other hand, if the new virus is already in there before the interferon binds, too bad, that second cell is toast as well, but that second cell will also be making interferon. So to recap, interferon is produced by virus-infected cells, especially our white blood cells. Then it binds to receptors of non-infected cell to prevent viral replication in that second cell. But it does nothing to help the first cell. We'll have a day in memory of all the first cells that gave up their lives. In addition to this, interferons will also activate natural killer cells to this virus and macrophages. And so what happens is, when it's releasing this, if there are other cells that contain this virus, the natural killer cells and the macrophages can go engulf those infected cells before those infected cells can release their thousands of new viral particles. So that's a good thing. As an aside, interferon-activated natural killer cells, basically natural killer cells also destroy malignant cells, so they're actually not going to be just targeting cells infected with viruses, but basically they're going to be targeting things that could help you. And I want to talk about this for a second so you understand how this works. Not that it's a details that are super important. But it brings us to what we call natural killer cell immunotherapy, which has been talked about a lot for the last year. So here's an example of what happens. If we talked about natural killer cells here being in blue, they can actually attack and destroy, in this case, melanoma tumor cells. Because this immunotherapy is used a lot for melanoma with great success. This is what cured President Carter's, I think he had stage 4 melanoma and it actually cured him. It's also used for pancreatic cancer. This is what Alex Trebek used very successfully, but it did not work in Supreme Court Justice Ruth Bader Ginsburg. So it's not for everybody, but you don't know until you get into a trial whether or not it will work for you. So here's why it sometimes doesn't work, because tumor cells, they figured out these natural killer cells can kill them, and they don't want that. So they can create a microenvironment that prevents these natural killer cells from doing their job, and thereby the tumor can survive, and not only survive, it can grow and metastasize and create havoc and kill you. So what was discovered is that if we can block autophagy in tumor cells, then those tumor cells will produce cytokines that will attract natural killer cells. So that prevents the tumor from escaping the effects of natural killer cells. And so these cytokines give this massive recruitment of natural killer cells, which are then going to kill the cancer cells, the tumors can shrink, and possibly you can be cured. All right, with that, we are done with our second line of defense. And we are completely done with all of our non-specific immunity. So come back for the next 2 Zoom videos as we learn about our specific